I want you to do something for me right now, just for a second. Bring your attention to the feeling of your clothes resting against your skin. Or, notice the exact pressure of the chair you're sitting on. Feel the way your shoes are kind of wrapping around your feet.
Yeah, you probably weren't feeling that a moment ago.
Right. A few seconds ago, you were completely oblivious to all of those physical sensations.
Yeah.
Your brain was entirely aggressively ignoring them.
It's a brilliant illusion, honestly. I mean, we navigate the day feeling like we're experiencing everything in our environment, right? Every sight, sound, touch.
Yeah.
But the reality of your physiology is that your nervous system is actively hiding almost the entire world from you at any given moment.
Which is wild. So welcome to this custom-tailored deep dive. We're unpacking a fascinating stack of source material today, covering the grand organization of the human nervous system.
The architecture of synapses, the incredibly wild world of neurotransmitters.
Exactly. We're going to explore how your body processes millions of bits of data every minute, how your brain filters all that noise, and how remarkably simple chemicals dictate every single thought, memory, and movement you make.
It's a lot to cover.
It is. Okay, let's unpack this, starting with that illusion. The fact that my brain was literally hiding my own clothes from me.
So that phenomenon is a core part of what we call the integrative function of the nervous system. And the numbers behind this are just staggering. Your brain actively discards more than 99% of all sensory information as totally irrelevant.
More than 99%.
More than 99%.
See, we tend to think of our minds as these giant sponges, right? Like they're meant to soak up every detail of our reality.
Right.
But they're operating much more like an incredibly ruthless bouncer at a club, just turning away almost everyone at the door.
That's a great way to put it, because think about the alternative. If that bouncer let everyone in, if your mind consciously processed every single touch of fabric, every microscopic shift in the room's temperature.
The hum of the fridge.
Exactly. You would be paralyzed by sensory overload.
Right.
So the nervous system only channels highly curated, important sensory information into the integrative and motor regions of the brain.
Okay, so it prioritizes.
Right. Like if you accidentally brush your hand against a hot stove, the nervous system instantly flags that tiny fraction of sensory data as critical. It bypasses the mundane chatter, channels it directly to the motor regions, and you pull your hand away before you even consciously register the heat.
Wow. Okay, so when I try to visualize this overall system, my mind immediately goes to like a decentralized network, maybe like a massive corporate hierarchy.
Yeah.
You have the sensory nerves gathering data out in the field. You have the brain acting as the executive board making all the massive strategic decisions.
Sure.
And the motor nerves are the workforce executing the orders. But in that model, I've always assumed the spinal cord is just the fiber optic cable.
Ah, right.
Just a fleshy data pipe plugging the local offices into headquarters.
That is a very common assumption. But I actually want to challenge that data pipe idea.
Oh, really?
Yeah. The spinal cord is highly organized. It's capable of executing incredibly complex computations all on its own without any input from headquarters.
Really? Without the brain?
Yeah, absolutely. In clinical scenarios where the spinal cord is completely disconnected from the brain, the neuronal circuits within the cord itself can still execute highly sophisticated functions.
Like what?
Well, they can orchestrate the complex, coordinated muscle movements required for walking.
Wait, the physical mechanics of walking are programmed directly into the spinal cord.
Yes. They trigger reflexes that stiffen your legs to support your body weight against gravity. They even control local blood vessel dilation and gastrointestinal pacing.
That dramatically shifts how I view the whole architecture.
I know, right?
Yeah.
The higher levels of the nervous system, the brain, they often don't bother micromanaging your legs. The brain simply sends a broad, general command signal down to the control centers in the spinal cord.
Like pushing a button?
Exactly. It's essentially telling the cord to just run its pre-programmed walking software.
That makes perfect sense when you look at the evolutionary heritage of the central nervous system.
Yeah.
I know the sources break this down into 3 major functional levels.
Right.
You've got that spinal cord level at the base, acting like local government,
Yeah.
handling all those automatic immediate reflexes.
Mm-hmm.
Then sitting just above that, you have the lower brain, or the subcortical level.
Which is the medulla, the pons, the thalamus, the hypothalamus. If the spinal cord is local government, this is regional management.
Okay.
It's where your most vital subconscious activities live.
Right, the subcortical level controls respiration, blood pressure, your sense of equilibrium.
Yeah.
But what really caught my attention in the material is that this lower brain also controls feeding reflexes.
Like salivation.
Yeah,
Yeah.
and even immense emotional patterns.
Yeah.
Anger, excitement, sexual response, pain, and pleasure. They are all generated here.
Yes.
And they can all occur even if the higher brain is completely removed from the equation.
Which brings us to the executive suite.
Yeah.
The higher brain, or the cortical level. The cerebral cortex.
The wrinkly part.
The wrinkly part, exactly. So if the lower brain is handling breathing and equilibrium and raw emotion, what does the massive cortex actually do?
Right.
Stripped down to its core, it is an extremely large memory storehouse. It's the database of your past experiences, your learned skills, and abstract thoughts.
But the cortex is totally dependent on the lower levels, isn't it?
It is.
The sources emphasize that while the cortex is essential for complex analytical thought, it can't function in a vacuum. The lower brain actually has to send signals up to wake up the cortex.
Right. It pries open its memory banks for the thinking machinery to use.
It's like the lower brain is the user actively booting up the hard drive of the higher brain.
That's a perfect analogy. Without that wake-up call from the lower brain, the cortex is essentially dormant.
Wow.
But, to truly understand how this massive three-tiered network communicates how it decides what to filter, what to ignore, and what to act on, we have to stop looking at the microscopic organs.
I have to zoom in.
Way, way in, down to the microscopic junction points.
Okay, so we are zooming in on a system that boasts an estimated 80 to 100 billion neurons.
Give or take a few billion, yeah.
Right. And the specialized location where any two of these neurons meet is called the synapse.
Exactly. And at these synapses, there are two fundamentally different ways neurons can pass information to each other, electrically and chemically.
Okay.
Electrical synapses physically connect the internal fluid, the cytoplasm, of two adjacent cells. They use these clusters of ion channels called gap junctions.
So the electrical signal just bridges the gap and flows right through from one cell to the next without translating into anything else.
Yep, it's a direct physical conduit. And because of that physical connection, electrical synapses are bidirectional.
Meaning it goes both ways.
Right, the electrical current can flow back and forth. That is incredibly useful if you need to perfectly synchronize a large group of neurons to fire at the exact same millisecond, like in certain muscle tissues.
But the material says that in the human central nervous system, the vast majority of synapses are chemical, not electrical.
Correct. Most are chemical.
And chemical synapses have one crucial architectural difference. They operate strictly as a one-way street.
Always one way. They only transmit signals from the neuron secreting the presynaptic neuron to the neuron receiving the postsynaptic neuron.
Why the limitation, though? I mean, if I'm building a massive information network, a bidirectional fiber optic link seems vastly superior to a one-way street.
You'd think so, right?
Yeah. Why rely on chemicals drifting across a gap?
Well, it really comes down to the difference between chaos and order. If signals could just bounce backward and forward indiscriminately across the network, the nervous system would be consumed by an echoing feedback loop of chaotic noise.
Oh, that makes sense.
The one-way chemical conduction forces signals to travel toward highly specific targeted destinations. It's what allows you to perform a discrete action like wiggling your left index finger without accidentally firing off the muscles in your shoulder.
Or like triggering a random childhood memory.
Exactly. Directionality creates precision.
Okay, let's visualize how this chemical handoff actually works.
Yeah.
Because the sheer scale and mechanics of it are mind-bending.
They really are.
So picture the very end of the first neuron, the presynaptic terminal. I kind of imagine it like a shipping port.
Mm-hmm.
Inside this terminal, there are thousands of tiny spherical containers called transmitter vesicles.
Mm-hmm.
And inside those vesicles is the chemical payload, the neurotransmitter.
And the trigger that releases that payload is an electrical signal, an action potential that arrives at the terminal.
Right. And when that electrical spark hits the terminal membrane, it alters the voltage, which forces open these specialized gates called voltage-gated calcium channels.
Yeah, calcium is the key player here.
Suddenly, calcium ions from outside the cell flood into the terminal. They act almost like microscopic dock workers, right? They physically bind to specific release sites on the inside of the membrane.
Which forces those spherical vesicles to fuse with the membrane and dump their chemical cargo outward into the gap.
And the amount of neurotransmitter dumped into that gap, the synaptic cleft, it's mathematically proportional to the exact amount of calcium that managed to enter the terminal.
Spot on, but you mentioned scale earlier. The scale of this environment is just impossible to fathom.
It really is. Like, take a specific transmitter, acetylcholine. A single one of those tiny vesicles contains between 2,000 and 10,000 individual molecules of acetylcholine.
That's just in one vesicle.
Yeah. And that synaptic cleft, the physical gap they were being dumped into, it's roughly 200 to 300 angstroms wide.
Tiny.
We are talking about a microscopic ocean of thousands of chemical molecules flooding across a canyon so unfathomably small it borders on the atomic scale.
It is a phenomenal biological currency exchange. So the chemical payload crosses the gap. Now it has to actually influence the second neuron, the postsynaptic membrane.
The receiving end.
Right. This receiving membrane is densely packed with receptor proteins. If the neurotransmitters are the keys, these receptors are the locks.
And these receptor locks are embedded right into the cell wall. They have a binding component pointing outward into the gap to catch the chemical key, and an intracellular component that reaches all the way through the membrane to the inside of the second neuron.
Exactly. And when a neurotransmitter key slots into the receptor lock, it can open the door in two very distinct ways.
Okay, what's the first way?
The first method is direct and lightning fast. These are called ionotropic receptors. When the neurotransmitter binds, the receptor physically shifts its shape and springs an ion channel open in a fraction of a millisecond.
Wow, that is fast.
Very. If it opens a channel that lets positive sodium ions rush in, it excites the neuron. If it opens a channel that lets negative chloride ions in, it inhibits the neuron.
Okay, but I have a fundamental problem with the second method then.
Oh.
Yeah. I mean, if the brain has evolved a mechanism to spring open these ion channels in a fraction of a millisecond, why bother with the other type?
The metabotropic receptors.
Right, the second messenger systems. The sources describe them, and they sound incredibly slow, complex, and biochemically expensive compared to just popping a door open. If survival is about speed, why build a slow system?
That's a great question, because survival isn't just about reacting to the present. It's about learning for the future. Speed is for the immediate reflex, right? Pulling your hand off the hot stove.
Yeah, that makes sense.
But what happens the moment the neurotransmitter detaches and floats away?
Well, the ion channel snaps shut instantly. The signal is gone.
Exactly. The electrical spark fades in milliseconds. So how do you retain a memory for 50 years?
Ooh.
How does your brain maintain a state of anxiety or focus for an entire afternoon? You cannot rely on a door that snaps shut instantly.
You need something that sticks around.
You need a system that translates a brief chemical signal into prolonged structural changes inside the cell. That is the genius of the second messenger system. And the most prominent player here is something called a G protein.
Okay, let's walk through the mechanics of the G protein, because honestly, this genuinely felt like the Rosetta Stone for understanding neuroplasticity.
It really is. Picture the G protein as a dormant, three-part molecular machine attached to the inside of the receptor lock.
Oh.
It has an alpha, a beta, and a gamma subunit. Normally, it just sits there completely inactive. But when a neurotransmitter binds to the outside of the receptor, the entire receptor complex alters its shape.
And that internal shifting does what?
It causes the alpha subunit of the G protein to grab a high-energy molecule called GTP. Energized by this, the alpha subunit physically breaks off from the rest of the complex.
So you now have this activated alpha subunit operating as an independent roaming agent inside the receiving neuron.
Yes, and this roaming alpha subunit is incredibly versatile. It can wedge itself into an ion channel from the inside and prop it open for minutes or hours rather than milliseconds. It can activate internal enzymes that set off massive cascading chemical reactions throughout the entire cell. Or, most profoundly, it can migrate deep into the nucleus of the neuron and initiate gene transcription.
Gene transcription. Meaning this little roaming subunit actually interfaces with the neuron's DNA.
Yes.
And it instructs it to start manufacturing new proteins, physically altering the structural architecture of the neuron itself.
Exactly.
That realization was just profound for me. A memory isn't just a lingering spark of electricity floating in the ether. The neuron is literally tearing down and rebuilding its own physical scaffolding.
It is cellular remodeling. And it is all triggered by a single chemical key drifting across a microscopic gap.
Incredible. Okay, here's where it gets really interesting. We've mapped out how the locks work, but let's examine the keys. The chemical cocktail itself.
Right.
Researchers have identified over 50 different neurotransmitters operating in the nervous system.
And functionally, they are divided into two main categories. You have small molecule transmitters and neuropeptides.
I like to think of them as the sprinters and the marathon runners. The small molecule transmitters are the sprinters. They're built for speed, they cause acute electrical responses, and they are highly efficient because they are recycled right there at the synapse.
Yep.
Acetylcholine is a classic sprinter, usually sending an excitatory signal. Dopamine is another, though it's typically inhibitory. Glycine is a sprinter that always acts as an inhibitor, particularly in the spinal cord.
And then we have GABA, gamma-aminobutyric acid.
Right.
The source material highlights a staggering paradox regarding GABA. In a fully developed adult central nervous system, GABA is the premier inhibitory neurotransmitter. It is the chemical brake pedal.
Okay.
But during embryonic development and continuing through the very first week of a newborn's life, GABA behaves as an excitatory transmitter.
That is just wild. The precise same chemical molecule performing the exact opposite physiological function just because the brain is at a different stage of life.
Isn't that amazing? It reveals something fundamental about how our brains wire themselves. An embryonic brain doesn't need brakes.
Right, it needs to grow.
It needs massive, relentless electrical activity to stimulate growth and form trillions of new connections. The GABA molecule itself doesn't possess inherent excitatory or inhibitory magic.
Oh, I see.
Its function is entirely dictated by the specific receptors and internal ion concentrations of the postsynaptic neuron, which change as the brain matures.
So cool. Another sprinter that completely breaks the standard mold is nitric oxide.
Ah, yes!
The rule breaker.
First of all, it's a gas.
Yeah.
And secondly, it's not stored in those little vesicles waiting for a calcium trigger.
Right, nitric oxide ignores the normal shipping infrastructure entirely. It is synthesized by the neuron instantly, strictly on demand.
Wow.
And because it's a gas, it isn't released into the synaptic cleft in discrete packets. It simply diffuses out of the terminal in all directions. It penetrates the membranes of dozens of neighboring cells to modify their metabolic functions for seconds or minutes at a time.
Just a cloud of influence.
Exactly. It's
Okay.
deeply implicated in the long-term shifts in behavior and memory formation. So those are the sprinters. Now for the marathon runners, the neuropeptides. The sources explain that the manufacturing process for these is entirely different and much more grueling.
Oh, absolutely. Neuropeptides are not synthesized at the terminal end. They are painstakingly assembled deep within the main body of the cell. They're manufactured by the ribosomes and packaged by the Golgi apparatus.
Way back at headquarters.
Right. Once packaged into vesicles, they have to be physically transported all the way down the long axon of the neuron to the terminal.
And the transportation speed is agonizingly slow. The material states they move at a rate of only a few centimeters a day.
A day.
Because the logistics of creating and shipping them are so intensive, they are released in minute quantities compared to the sprinters.
But they compensate for that low volume with sheer power. Neuropeptides are generally 1,000 times more potent than small molecule transmitters.
1,000 times.
Yeah. And their effects aren't measured in milliseconds. They cause prolonged actions that can alter a cell's function for days, months, or even years.
Okay, the system gets even more complex, though, because a neuron doesn't have to choose between being a sprinter or a marathon runner. The text details a phenomenon called co-release.
Right, co-release. A single presynaptic neuron can store and simultaneously release both a fast-acting small molecule and a slow, potent neuropeptide
At the same time.
at the exact same time. For example, certain neurons in the Rocropho nucleus of the brainstem will co-release serotonin alongside glutamate.
Okay.
This dual action allows the neuron to hit its target with a rapid, immediate instruction, a quick voltage spike followed immediately by a slow, long-term metabolic adjustment.
Okay, let's pause and integrate all of this,
Yeah,
because my head is spinning a little.
it's a lot of moving parts.
Picture a single receiving neuron. Across its surface, it has tens of thousands of these presynaptic terminals docked to it.
Mm-hmm.
At any given millisecond, they are raining down dozens of different chemicals. Sprinters, marathon runners, excitatory keys opening sodium channels, inhibitory keys opening chloride channels.
Total chaos.
Exactly. How does this single neuron sort through all that noise and decide whether or not to fire its own signal?
It executes a continuous process of electrical math.
Math.
Yeah. To understand the math, look at the neuron's baseline resting state. Inside the main body of a resting neuron, the electrical potential is heavily negative, about negative 65 millivolts.
Negative 65.
Ah.
And it uses biological pumps to maintain that negativity because, based on the physics of Nernst potentials, the surrounding environment is super hostile.
Very hostile.
Sodium ions are highly concentrated outside the cell, and because they are positively charged, physical forces are desperately trying to shove them inside. Meanwhile, potassium is concentrated inside and wants out, and negative chloride wants in.
Exactly. The resting neuron is holding back immense chemical and electrical pressure. Now imagine an excitatory neurotransmitter binds to a receptor and pops open a sodium channel.
Okay.
Positive sodium ions rush inside. This influx of positive charge pushes the internal voltage upward, neutralizing some of the negativity, moving it from negative 65 millivolts towards, say, negative 45 millivolts.
Okay.
That upward positive shift is an excitatory postsynaptic potential, or EPSP.
Conversely, if an inhibitory transmitter binds and opens a chloride channel, negative chloride rushes in. Or it opens a potassium channel and positive charge bleeds out.
Right.
Either way, the interior becomes even more negative, dropping to perhaps negative 70 millivolts. That is an inhibitory postsynaptic potential, or IPSP.
So moment to moment, the neuron is undulating with these tiny upward EPSPs and downward IPSPs.
I picture the neuron as a massive town hall meeting. The threshold to actually take action to fire an action potential down its own axon requires the internal voltage to rise about 20 millivolts above resting.
Correct.
But a single synapse firing only shifts the voltage by 0.5 to 1 millivolt. One single synapse screaming fire isn't enough to pass the motion. You need summation.
Yes, summation is how the neuron tallies all those votes. Spatial summation occurs when 40 to 80 different terminals, scattered all across the vast, branching dendrites of the cell, all vote excite at the exact same moment.
Their tiny individual voltage changes pool together.
Exactly. And temporal summation is when one highly active terminal votes excite over and over in rapid-fire succession.
Stacking its own voltage changes on top of each other before the previous one has time to fade.
You got it. But beyond just the sheer number of votes, there is a strict geographical hierarchy to this process.
Oh, right, the leaky dendrites.
Yeah, the dendrites, the long branches reaching out to receive signals, are electrically leaky. They lack sufficient voltage-gated sodium channels to sustain a signal perfectly.
Okay.
As a tiny electrical voltage travels down the length of the dendrite toward the main cell body, it dissipates. This is known as decremental conduction.
Which means the physical architecture dictates the logic. A synapse located all the way out at the distant tip of a dendrite has significantly less voting power than a synapse docked right next to the main cell body.
Because its signal just leaks away during transit. Geography is destiny in the neuron.
Wow.
The ultimate tallying center, the location where the final vote is counted, is the initial segment of the axon, right where it emerges from the cell body.
The axon hillock.
The axon hillock. It possesses 7 times the concentration of voltage-gated sodium channels compared to the rest of the cell. If the summated voltage reaches the critical threshold at this specific location, the action potential is triggered and the neuron fires.
The precision of this system is breathtaking. The chemical voting, the competing electrical forces, the physical geography of the synapses. But biology isn't perfectly sterile, right?
Definitely not.
What happens when the chemical environment around this delicate town hall gets altered? For instance, what if a neuron is just aggressively stimulated nonstop? Does it just fire continuously until it dies?
Actually, it doesn't, because it undergoes synaptic fatigue. The presynaptic neuron literally exhausts its supply of transmitter vesicles.
Oh.
The shipping port runs entirely out of chemical cargo, and the signaling grinds to a halt.
At first glance, that sounds like a massive biological design flaw, like a system that easily depletes its own ammunition.
It seems like a flaw until you realize it is a vital, life-saving protective mechanism. Well, when regions of the brain become dangerously overexcited, such as during the onset of an epileptic seizure, it is primarily synaptic fatigue that steps in to subdue the runaway excitability.
Oh, wow.
Yeah, the neurons exhaust their neurotransmitters, forcing the seizure to break. The brain essentially mandates a temporary shutdown to protect its own tissue from burning out.
That is incredible. The brain is also shockingly sensitive to the pH level of its surrounding fluids, isn't it?
Very sensitive.
The sources mentioned normal blood pH sits tightly at 7.4. If that rises into alkalosis, neuronal excitability absolutely skyrockets. Something as seemingly benign as hyperventilating, which blows off excess carbon dioxide and raises your pH, can elevate excitability enough to trigger a severe epileptic attack in individuals who are predisposed.
And we see the exact inverse with acidosis. If the pH dips below 7.0, neuronal activity is profoundly depressed.
Meaning?
This mechanism is why conditions like severe diabetic acidosis almost universally plunge the patient into a comatose state. The neurons simply become too suppressed to function.
Then you factor in oxygen dependency. Interrupting blood flow to the brain for merely 3 to 7 seconds results in complete unconsciousness.
Yep.
And of course, there are external chemicals hacking the system. Caffeine lowers the threshold for excitation, making it much easier for the axon hillock to reach the voting threshold and fire.
Right.
But the detail that really stuck with me is how strychnine works. Strychnine poisoning causes severe, often lethal muscle spasms. But it doesn't achieve this by stimulating the neurons into a frenzy.
No, the mechanism of strychnine is terrifyingly elegant. It doesn't press the gas pedal, it completely cuts the brakes.
Mhm.
Strychnine physically blocks the receptors for the inhibitory transmitter glycine, particularly in the spinal cord.
So without the brakes.
Without those constant downward IPSP votes keeping the network in check, the normal baseline excitatory chatter of the nervous system is left totally unopposed. It snowballs into massive, uncontrolled, repetitive discharges, overwhelming the motor neurons and causing those catastrophic spasms.
It all comes back to that razor-thin balance of competing forces.
Hmm.
So what does this all mean for you as you go about your day?
Yeah.
It means that every single action you take, the way you process a complex problem at work, the way you regulate your frustration in traffic, the way you recall the details of this deep dive, it all relies on this microscopic math. Billions of neurons constantly balancing the influx of sodium against the outflux of potassium, weighing sprinters against marathon runners.
It's a symphony.
And that 99% sensory filter we explored at the very beginning, that active suppression is the only reason you were able to focus your higher cortex on our conversation today without being overwhelmed by the physical sensation of the clothes on your back.
And here is a final reality to consider, derived directly from the mechanisms we've just explored.
Okay.
Because you have spent the last 15 minutes or so actively listening, focusing and processing this complex information, the G proteins within your postsynaptic neurons have been activated.
Oh my gosh.
The alpha subunits have broken off. Right now, at this very moment, gene transcription is occurring deep inside your brain. Your DNA is actively manufacturing new proteins, permanently altering the physical structural scaffolding of your neurons for the sole purpose of stirring the memory of this deep dive.
Your brain is physically, tangibly different right now than it was when you first pressed play. Thank you for joining us on this deep dive. Keep questioning the world around you, keep learning, and take a moment today to appreciate that incredibly bizarre, microscopic machinery constantly computing inside your own head